Hello biology buddies, and welcome to lesson 6 in the unit B1, so the cell biology unit for the AQA GCSE science. Your YouTube video is right here. Normally in the textbooks and often in class you'll do animal stem cells and plant stem cells together in the same lesson. I'm going to split them up into two different videos here. So lesson 6 is animal stem cells and differentiation, and lesson 7 will be plant stem cells, so looking at meristem, plant cloning, things like that. For this lesson, we're just going to focus on animal stem cells. We're going to be able to recall what a stem cell is by the end of the lesson, describe the process of differentiation, also sometimes called cell specialization. We're going to talk about where stem cells are found in animals. We're going to be focusing on humans. We're going to explain the process of therapeutic cloning, so what is therapeutic cloning, what's it's used for, and then we're going to evaluate the use of therapeutic cloning and get into some ethics as we do that. Let's start off really simply as to what a stem cell is. A stem cell is an unspecialized cell that can divide and differentiate into specialized cells. When we say divide, we mean by mitosis. There's a lesson on the cell cycle and mitosis that's coming up after the animal stem cell and plant stem cell stuff, and it can differentiate into specialized cells, and that was an earlier lesson. So specialized cells, for example, sperm, neuron, root hair cell in plants, muscle cell in animals, humans, palisade cell, xylem, phloem, all that good stuff. So an unspecialized cell turning into a specialized cell, it's only stem cells that are able to do that. The process by which the stem cell turns into the specialized cell is called differentiation, also often called cell specialization. So if you see those two terms, for the sake of the GCSE, they are interchangeable. And differentiation involves the cell turning on or off different genes and then changing its structure to match its function. We talk all the time in biology about matching structure to function. A specialized cell has a specific job, a specific function within the organism, and it needs a structure to do that. So for example, the neuron has an elongated cytoplasm. It has lots of branching points at the soma, the cell body end, but also the axon terminal end. If we look at plants, a root hair cell has the elongated bit that sticks out into the soil to increase the surface area, etc., etc. So going from your unspecialized cell, your stem cell, to a specialized cell is going to involve not just changes in terms of what genes are being expressed, but also the shape and the structure of the cell itself. So that's going to apply to both animal cells and plant cells. We're going to look at animal cell specific stuff now. And for that, we're going to talk about two different types of stem cells. You've got embryonic stem cells and you've got adult stem cells. Embryonic stem cells are found in the very early embryo. So if you have a sperm and egg that combine together to make a zygote, that divides to make 2 cells. Those are embryonic stem cells. Those divide to make 8 cells. Those are all embryonic stem cells. Those divide to make, 4 and then 8. Those are all embryonic stem cells. And if you think about it, you come from one cell. So the sperm from your father, the egg from your mother, fusing together through the process of fertilization. That one cell, that zygote, is your beginning. That zygote then splits to make more cells through the process of mitosis. But those early cells have to be embryonic stem cells because they have to be able to turn into, to differentiate into, all of the specialized cells that make up your body. Your muscles, your bones, your nerves, all the different organs in your digestive system, all the stuff in your nervous system. So loads and loads of different types of cells all come from those original cells. So those original cells have to be embryonic stem cells. Some animals keep embryonic stem cells with them for their entire lives. Unfortunately, humans don't do that. So after the very early embryo, we don't have any more embryonic stem cells. A baby does not have any embryonic stem cells. The beautiful thing about these, as we just alluded to, is they can differentiate to become any type of specialized cell. So again, You started life as one cell, the zygote, which divided to start to make the early embryo. Those cells are what we call totipotent. They can turn into any type of specialized cell. They can differentiate into any type of specialized cell. And that's what makes embryonic stem cells so special. Adult stem cells, on the other hand, and don't let the name fool you, babies have adult stem cells. A fetus, before the baby is born, has adult stem cells. A child has adult stem cells. Adult stem cells can only turn into a few different specialized cells. So they don't have the superpower that embryonic stem cells do to be able to differentiate into any type of specialized cell. They can only differentiate into some specific ones. The example that they love in the GCSE is bone marrow. So you have bone marrow. If you cracked open one of your large bones and looked down the middle, you would have a squishy substance on the inside. Those of you who enjoy eating fancy food, you can get bone marrow from different animals to add to your food. Very, very rich in taste. Try it if you are of the want to do so. But that stuff is adult stem cells, or it contains adult stem cells, but because it's adult stem cells, it cannot turn into every type of cell. It can only differentiate to become some. So bone marrow can become blood cells, so your red blood cells, some of your white blood cells, things like that. It cannot become neurons, it cannot become muscle cells. It can turn into some cells, it can differentiate into some specialized cells, but not all of them. So big difference there. Embryonic stem cell can differentiate into any specialized cell. It has that superpower. Adult stem cells can only differentiate into a few different types of specialized cell. And the example you have to remember is bone marrow, which can differentiate to become blood cells. We often talk about, and often comes up on the exam, the idea of using stem cells in medicine. So this is where the idea of therapeutic, from the therapeutic cloning, is going to come from, and we'll get to that in a minute. But a therapy is sort of a medical intervention that's going to help somebody. So how could we use stem cells? Well, stem cells can differentiate to become other types of cells. So if you need those other types of cells, or the tissues or organs that they work together to make, then in theory you could use stem cells for that. People with sickle cell anemia, that's a problem with red blood cells. You could be cured through a bone marrow transplant, so getting bone marrow from somebody else. Bone marrow contains adult stem cells. Those adult stem cells can divide through mitosis and then differentiate to become red blood cells. So if you have a problem with your red blood cells, getting somebody else's bone marrow can potentially treat or even cure that condition. That's a case of using adult stem cells. Extraction of bone marrow can be painful. So when we talk about advantages and disadvantages of different uses of stem cells, one negative of using adult stem cells, beyond the fact that it can only differentiate to become a few different types of cells, is that it can be painful to extract the bone marrow. It's not life-changing pain, but it wouldn't be that pleasant. What about embryonic stem cells? Embryonic stem cells, for a while, were considered sort of the holy grail of modern medicine. The idea being that if you had some embryonic stem cells, you could get them to turn into any type of cell and therefore work together to make any type of tissue or organ in the body. So being able to cure diseases or fix massive injuries that in the past would have been life-threatening or at the very least life-altering. So for example, Parkinson's is a really, really nasty disease of the nervous system. It affects nerve cells. In theory, you could use embryonic stem cells to repair and regrow nerve cells or neurons to cure somebody who has Parkinson's. Somebody who has type 1 diabetes, in Unit B5 we talk about diabetes. Type 1 diabetes is a problem with some of the cells in the pancreas that don't produce insulin or don't produce enough insulin. If you had embryonic stem cells at your disposal and you were able to turn or differentiate those stem cells into those cells in the pancreas, then you could cure somebody of their diabetes because you could give them functioning pancreatic cells. You could also do things to repair damage. So somebody who's had a spinal cord injury, if you sever the spinal cord in Unit B6, we learned that the spinal cord is part of the central nervous system along with the brain. If you have a complete sever of your spinal cord, everything below that break, you will not be able to feel or have any control over. So people who are in really traumatic car accidents or other sorts of accidents where they break their back and sever their spinal cord, that's it, they're paralyzed. In theory, you could repair that damage using embryonic stem cells by regrowing the neurons that make up that spinal cord. Potentially even create new organs. So create a new heart for somebody who has heart problems. If they had some heart damage, in theory, you could repair that damage with embryonic stem cells by getting the embryonic stem cells to differentiate into cardiac, that's heart cells, and repair the damage that way. Or if it was a really bad situation, potentially even grow them a new organ using embryonic stem cells. Same thing for the eye, same thing for the liver. Liver is super important. If you have problems with your liver, it can be life-threatening. So embryonic stem cells, massive, massive deal if we can get it to work properly in medicine, because you can take those stem cells and differentiate them into any type of specialized cell. You may be saying to yourself, well, why do we need to have grow a new heart, or grow a new eye, grow a new liver? Surely we have organ transplants, and that's true, there are organ transplants. There's two main problems though with organ transplants. Number one, there's nowhere near enough donor organs to meet demand. So all around the world there are people on waiting lists for things like kidneys, lungs, heart, liver, any organ you can think of. There's people who are waiting for one and they're on a list, and if one doesn't become available, eventually they will die. The other problem is what's called organ rejection. We've probably heard about organ rejection. So the first problem, there's not enough supply to meet the demand. Second problem is organ rejection. And organ rejection is because your own immune system will recognize the organ as not being you, and therefore will attack it and destroy it. So that's why whenever somebody's trying to get an organ, they have to find somebody who is quote unquote a match. And that's someone who is as close as possible to them so that the immune system will not attack it as quickly right away. It's a massive oversimplification. But the closer the organ is to you, the less likely or vigorously your immune system is to attack it. So people who get organ transplants, even if it's from a family member, unless it's an identical twin, they're going to have to go on what's called immunosuppressants. That's not how you spell suppressants, but that's okay, we get the point. And an immunosuppressant is literally something that weakens the immune system. So somebody who, spelling is atrocious today, somebody who gets an organ transplant, great, to potentially save their life, but they're going to need to take these drugs that actually make their immune system weaker, which means that they're much more susceptible to catching other diseases because their immune system is not strong enough to fight it off. And you learn more about how the immune system works in Unit B3. Now we move on to therapeutic cloning, and there's some key words that we need to remember. The first is clone. Super, super, super important idea. It comes up in B1, in B2, in B6. Clone is really, really important. Very simple definition. A genetically identical organism. A genetically identical organism. That's a really simple recall question that's going to come up on the exams. 1 mark. What is a clone? You just write those 3 words. Genetically identical organism. You get the mark. Therapy, as we said before, is a medical treatment that's used to fix a problem. So therapeutic cloning is creating clones for their use in medicine. Okay? So we're going to clone a patient. We're going to make a clone of them, but we're not going to allow it to grow and turn into a person. Right? You're not going to have a copy of yourself walking around. You are going to, and it's a bit of a gruesome word, harvest the embryonic stem cells. So early on, when you make the clone, you have that one cell, which is the zygote, which divides to make 2, which divides to make 4, which divides to make 8. You would stop the clone from growing at this stage and take those embryonic stem cells. And now you have embryonic stem cells that are genetically identical to you. Okay? because you're cloning yourself, those embryonic stem cells would be genetically identical to the patient. Which, as we'll see, solves one of the problems of organ transplants. So problem one that we said was there's not enough donors of organs. Well, you don't have to wait around if you're going to do therapeutic cloning, because in theory, you make a clone of yourself, you harvest the embryonic stem cells, and you can grow yourself an organ, or you can use those embryonic stem cells to heal or repair the organs that you already have. So it solves problem number one. Problem number two, it solves as well, because we said for organ rejection that it's because the immune system doesn't recognize the organ as being part of you. It thinks it's something foreign, it's from the outside. Well, if it's a clone of you, then your organ will be genetically identical. So your immune system won't think that it's an outsider, won't think that it's a foreign thing that needs to be destroyed. It will see it as part of you, so it will not attack it. So in theory, therapeutic cloning, fantastic. Make a clone, harvest the embryonic stem cells, don't have to worry about organ transplants, don't have to worry about donors, don't have to worry about immunosuppressants. Win, win, win. There is still one potential thing that can come up, which we'll go to here, which is viruses. If there's a viral infection, so if there's a virus that has gone in, and viruses come up in unit B3, they work by getting inside the cell and tricking the cell into mass producing more of the virus. So if you had an embryonic stem cell with a virus inside of it, and then you use that to help cure the person, you would be giving that person the virus. Okay, if you transplanted an organ in and the cells that make up that organ were infected with a virus, when you put that organ into the person, you would be infecting them with a virus. Okay? So one potential issue with doing it this way. So we've talked about the benefits of therapeutic cloning. We've talked about one potential disadvantage, which is a viral infection in the stem cell. But how could it be done? And in theory, this is super simple. And in fact, if you think of animals like Dolly the sheep, or Snuppy the puppy, or any of the other animals who have been cloned, they use this basic system. We're not going to get into all the details here, but we'll go through the basic steps. And the way that this sort of cloning is done is called nuclear transfer cloning, because what you're doing is taking the nucleus from one cell and putting it into another cell. So what would we do? We would take a body cell from the patient, so anything other than a sex cell. There's my cell. I've got the nucleus in there, and I'm going to remove the nucleus from the cell. So I've got my nucleus here, and now I have an empty cell. I get rid of that empty cell. I'm then going to take an egg cell, and I'm going to take the nucleus from that egg cell, and I'm going to remove it. And I'm going to get rid of that nucleus. So I end up with an empty egg cell. Then I take the nucleus from the body cell, so from step one, and I put it into the empty egg cell. And what I've created is essentially a fertilized egg. Why is it a fertilized egg? Because it has a full set of chromosomes. A sperm cell and an egg cell have half the number of chromosomes. So an egg cell on its own cannot become a person. A sperm cell on its own cannot become a person because it only has half the chromosomes that person would need. But I've got the full set of chromosomes because I've taken it from the person's body cell. And a body cell has the full set of chromosomes. So in a typical human being, that's 46 chromosomes or 23 pairs. You get 23 from dad through the sperm, 23 from mom through the egg. When you have a full set of chromosomes in an egg, that's essentially a fertilized egg. So I've created a fertilized egg that is genetically identical to the original patient. Then, and this is the bit that always I find fascinating, you give it a small electric shock. And the reason that I find that fascinating is because if you think back to Frankenstein, I know you've all read Frankenstein, Mary Shelley's masterpiece, the way that the scientist, Dr. Frankenstein, the monster is actually called Frankenstein's monster. It's not called Frankenstein. The way that Dr. Frankenstein brings his monster to life is by harnessing the power of lightning, so electricity. So we're doing a very similar thing here. We give, and there's my lightning bolt. I know that it's obvious that it's a lightning bolt. We give it a little electric shock. We're not going to hook it up to a giant contraption to harness lightning into it like they do in the book Frankenstein. We give it a little electric shock, and now that cell is going to start dividing just like an embryo. So it's going to divide, and really importantly, when it divides by mitosis, it produces genetically identical copies. That's going to continue dividing. We end up with our 4, then we end up with our 8, etc., etc. Each of those is genetically identical. That's step 5, and each of those cells is an embryonic stem cell. I then break up those cells and take them for therapeutic use. And each of those, again, is an embryonic stem cell that in theory I could use to repair damaged organs or potentially even regrow organs from scratch. So looking back at what we've talked about from therapeutic cloning, therapeutic cloning could conceivably solve major problems with organ transplants, namely that you don't have to wait around for a donor, and there's no risk of organ rejection because you are creating an organ from your own clone. So it's going to be genetically identical. Your immune system is not going to recognize it as different. We talked about how there's a small problem with a potential viral infection in the embryonic stem cell, and we've looked how actually it's not that difficult to do. There are some technical challenges involved, but from a very basic standpoint, we can do it in 6 steps. Right? 6 steps to get embryonic stem cells. You may think to yourself, okay, well then what's the problem? This is fantastic. Let's do it. Comes down to some problems with ethics. And what are ethics? Ethics are the moral principles, and there's an obvious spelling mistake here, that govern a person's behavior. Okay? Your ethics will be shaped by the culture you grew up in, the family and friends that you surround yourselves by, but ultimately your ethics are your own moral principles. What do you think is the right thing to do and the wrong thing to do? For example, are you somebody who thinks that it's okay to steal no matter what? Are you somebody who thinks it's never okay to steal no matter the circumstances? Are you somebody who thinks it's wrong to steal, but if it's something like stealing a loaf of bread to feed your starving family, then that's okay? Those sorts of questions are ethical questions. They are moral questions. If you get a question about ethics on the exam, it's really important to remember that there's no right or wrong answer. The examiner is not going to read your answer and go, well, I have a different set of beliefs than this person, so I'm going to mark them as wrong. As long as you present positives and negatives and then make a conclusion, and that your positive and negatives make sense, they're not just, well, the space alien from Mars told me X, Y, and Z, then you're going to get the marks. Let's do a quick ethics example. And if you're interested in ethics and you think to yourself, well, it's a cool hobby, there are jobs involved with ethics and morality. So for example, working at a hospital and being part of the team who decides who gets an organ. Remember, there's not enough organs for everybody, so you need to decide who gets an organ. How is the money for the hospital spent? There's only so much money, so some people are going to benefit, some people aren't. A lot of those are moral, ethical questions. So let's do an example problem together. This is called the trolley problem, and I'm going to do a beautiful picture for you here. I'm going to draw some train tracks. And then here the train tracks go down like that. And we're going to put 5 people standing over here. Absolutely not paying attention. And then we have 1 person standing down here on these train tracks. Let's keep these going. So you have 5 fools standing on the train tracks over on this end, and you have 1 fool standing on the train tracks over on this end. You also, unfortunately, have a beautifully drawn train. There's the steering wheel with somebody who, those are little X's for eyes, the driver has died or is unconscious and is unable to control the train. So this train is zipping along, going that way, and it's about to run into these 5 people, killing them all. Oh no, what can you do? Well, luckily, you are standing over here far enough away that none of these people can hear you or see you, but close enough that you can see what's going on, and you have a lever which controls the switch on the tracks. So you have a decision that you need to make. Are you going to, there's you trying to make up your mind, are you going to throw the lever, which will then change the track that the train is on and make it go down here and kill the person? Or will you leave the lever alone and allow the train to go through and kill these 5 people? There is nothing you can do to stop the train from killing somebody. It's either going to kill 5 people, or if you throw the switch, then it's going to kill 1 person. What are you going to do? I'd like you to pause the video and actually make a decision. You need to say, I pull the lever, kill the 1 person, or I leave the lever alone and the 5 people die. I'm going to give you 10 seconds now to come up with your own decision, and then I'm going to tell you that you're a terrible person. Okay, so hopefully you've made a decision. Let's talk through the first possible scenario where you decide, I do not want to kill this person over here. I am not a murderer. I do not want to be responsible for their death. It's not my fault that the train's going along, so I'm going to let the 5 people die. So if you don't throw the switch, you let those 5 people die, and you can argue to yourself, well, I have not directly caused the death of anybody. I haven't murdered anybody, so it's okay with me. It's their own fault for being on the train there. And my response to you would be, you had the opportunity to save 5 people's lives, and all you had to do was throw a switch, and you were too lazy or too inconsiderate to even do that. You monster. Let that sink in. You are a monster. You let 5 people die because you couldn't be bothered to throw a switch. Okay, now let's talk about the people who said that they would throw the switch. You are a murderer. You have directly caused the death of another human being. Let that sink in. You may have thought to yourself, I wouldn't kill somebody. Well, you did. You've never met this person. They've never done anything wrong to you. They've never posed a danger to anybody else. They weren't threatening anybody. You just killed a complete stranger that you've never met. You are a monster. Now, some of you might argue the good of the many outweighs the good of the few. Okay, so now you're talking about different moral positions. You're talking about things like utilitarianism. Some of you may say, I would throw the switch and kill the person. Okay, would you still kill the person if it was your boyfriend or girlfriend? Would you still throw the switch and kill the person to save 5 others if it was a parent or a brother or a sister or somebody you cared deeply about? There are no right or wrong decisions here. There are only bad and bad decisions. And you need to be able to pick which one you want to do. Okay, so hopefully you feel terrible about yourself and you think that you're a complete monster and you go, why is Mr. Craven doing this? Well, it does have to do with the use of embryonic stem cells. In England, an embryo is not legally recognized as a person. Okay? That's why abortion at certain stages is legal. You do not get arrested for murder. You have not killed a person. Under the law, the embryo is not a person. An embryo does, however, have the potential to become a person if it were allowed to develop into a fetus and then born as a baby. Right? So even though an embryo is not recognized legally as a person in England, it could, if you left it alone, it wouldn't guarantee, there's things that can go wrong, but it could develop and become a person. When you harvest stem cells from an embryo, so as part of therapeutic cloning, that we all said before was so fantastic, you destroy the embryo. Now you're not killing a person because it's not a person yet, but you are preventing a person from even ever being born. So it is a moral decision. It is a moral question for individuals and societies to come together and decide, is it acceptable for us to create these embryos and then destroy them, which prevents the possibility of that person being born, in order to use those cells and treat and cure other people who are alive right now. There is not a right answer to that. There is not a wrong answer to that. That is a moral position that everybody needs to decide for themselves. So when we say that there's ethical problems with therapeutic cloning, with using embryos for research, it's because in the process you are destroying the embryo. And while you are not killing a person, you are preventing a potential person from being born. Okay? So we talked about evaluate questions before. We talked about how it doesn't matter what the moral, religious, cultural background your examiner has. They need to mark it in a certain way. They're going to be looking for arguments for something, arguments against something, and a conclusion. That conclusion needs to reference that and that. Often people say, I don't know how to write a conclusion. I'm just going to give up on that 1 mark on the exam. I'll tell you, it's really, really easy to write a conclusion. And the way you do it is you start by saying, in conclusion, cannot write today, in conclusion, and then let the rest of the words come out. Okay? And you get your mark. So, how would I prepare this exam question? I think for a 6-mark question, it's always good to have a little plan. You can do it in the margin. It's going to take you 30 seconds or so to do. And if you make a little box like this and you put in something from each box, you're going to get full marks on this question, provided you also put in a conclusion. So if the question was, evaluate the use of embryonic stem cells and adult stem cells in medicine. Bunch of things to note here. First off, evaluate means I need to put advantage, disadvantage, conclusion. I need to talk about embryonic stem cells and adult stem cells. A number of people who do a question like this and are going to say, I'm just going to talk about embryonic stem cells, or I'm just going to talk about adult stem cells, you're not going to get full marks. So make yourself a little table like this. Have a think to yourself right now. Pause the video. What are the advantages that I could put in for embryonic and adult stem cells? What are the disadvantages I could put in? Here are the basic ones that I came up with. So advantages of using embryonic stem cells is, as we've said, repeatedly, they can differentiate to become any type of specialized cell. So they can be used in medicine for a lot more things than adult stem cells can. Advantage of adult stem cell, nothing is being destroyed, right? If you are taking some of somebody's bone marrow, you are not killing that person. You are not preventing that person from having been born. You're not preventing that person from having children in the future. It's just going to be a little bit painful for them. So nothing is getting killed. Nothing is getting destroyed. Disadvantage of embryonic stem cells obviously is that it destroys the embryo. And you can talk about how that leads to ethical concerns because some people don't like the idea of destroying embryos. Adult stem cell, biggest disadvantage is it can only differentiate to become some specialized cells. So you're limited in what you can use those cells for. Now, GCSE science is way behind actual science in modern medicine. Okay? The actual possibilities here are a lot more complicated than what comes up on the exam. You don't really want to talk about this too much on the exam. You want to focus on the specification. But for your own knowledge, there's lots of research that suggests we can actually take adult stem cells and trick them into thinking they're embryonic stem cells. So we can turn adult stem cells into embryonic stem cells. Now that would be amazing because it would eliminate the problems associated with therapeutic cloning. You would no longer have to create embryos just to destroy them and get the stem cells. If you could take adult stem cells or even specialized cells and turn them by switching on and off their genes back into embryonic stem cells, that solves that problem completely and you don't have to worry about the morality and ethics of it. Okay? But for the GCSE AQA, these are the advantages and disadvantages that we need to focus on. Okay, that's it for this lesson. We've talked about what a stem cell is. We need to remember it's an unspecialized cell that can differentiate into specialized cells. Process of differentiation is how the stem cell becomes a specialized cell. And here we're talking about different genes being turned on or off and changes in structure, which will lead to changes in function. Describe where stem cells are found in animals. We talked about how you have to know about embryonic stem cells and you have to know about adult stem cells. Embryonic stem cells, as the name suggests, is found in the embryo, whereas adult stem cells, an example, is in the bone marrow. Explain the process of therapeutic cloning. Therapeutic cloning, you're going to make a clone of the patient, and then you're going to harvest or extract the stem cells, which are embryonic. We talked about the advantages of that, particularly when it comes to organ transplants, not having to wait for an organ, not having to worry about organ rejection. And we talked about problems with that. Possibility of viral infection in the initial embryonic stem cell, which would mean that the organ that's being transplanted in would have the virus, so you're giving somebody the virus. But also all of the ethical concerns that come along with the creation of embryos just to harvest the stem cells and destroy them. Evaluate the use of therapeutic cloning. We need to remember advantages, disadvantages, and a conclusion. And that'll get you your 4 marks, your 5 marks, your 6 marks. Okay? Lovely to have you listen to this. We will do the plant stem cells next. Thank you so much, and please make sure to complete the forms. If you heard a rocking noise in the background or some weird baby sounds, I apologize for that. I literally have my newborn son sitting next to me in a chair. He is sleeping but talking a little bit, and I am rocking him, trying to keep him asleep with my foot, which is that noise in the background. If you didn't notice that, then just pretend I didn't say anything. Okay, see you soon, and keep enjoying biology. Bye-bye.